Welcome back to AP Psychology, and we're taking a look at research methods. This is Unit 2 in the Myers text. What does it mean to think like a scientist? And you'll see by the subtitle that I'm implying that intuition can be a bad thing. Scientists have a reputation for not relying on intuition so much as evidence. And that's the key idea, that intuition doesn't prove a thing. We need ironclad proof, evidence, before we accept something as being the truth. At the core of the scientific engine, so to speak, the scientific method is curiosity. It's literally driven by curiosity. Most learning that we care about is driven by curiosity. In the realm of the scientific method, we begin with theory. In my research, social cognitive theory was at the heart. And that theory, whatever it happens to be, generates hypotheses or educated guesses. And then we conduct experiments in a logical fashion. We'll talk more about that here in the next slide. We gather data, we analyze that data, and we develop conclusions. Those conclusions may lead us to revising our theory or revising hypotheses for the next round of experiments. The scientific method, again, is based on theory, accepted theory in the world of science, in this case psychological science, and we use that to generate predictions or hypotheses. We conduct our experiments. This is done in science, in psychology. Psychology is indeed a valid science. We observe and analyze the results, and again we come back to modifying the theory or creating a new theory if that's called for. Here's an example of scientific thinking gone bad, and it's hindsight bias. It's the idea that we should have known something all along after the event has taken place. You don't do well in the test and you tell yourself, oh, I should have known to study X, Y, or Z. Well, you did your best. There's no way that you could have known that X, Y, or Z could have been on that test. That's hindsight bias. This image is of 9/11, and in the aftermath of 9/11, a horrific tragedy. People were pointing fingers left and right at various officials saying, you should have known that these people, these men were plotting an attack on New York City. Well, it's easy to say after the event has already happened. That's hindsight bias. So let's come back again to this idea of what it means to be a scientist and to adopt a scientific attitude. At the core, curiosity. You look at the world and you simply wonder. I've told you the story of having taught small children and how their sense of wonder is endless. Besides the two stories I've already shared with you, a third one I remember coming from a fourth grade child, about 9 years old. This girl wanted to know, how did the lead get inside a pencil? Well, if I were to ask any of you to describe that process, you would find it to be somewhat difficult. She was looking at a common, ordinary thing and wondering with just abundant curiosity. Well, that's what drives the scientific attitude. The next pillar we're going to consider is skepticism, not believing on face value people's claims to truth. The image you see is of the amazing Randy. He's a professional magician, and he's had a long-standing challenge to practitioners of the paranormal, that if they can demonstrate their skills in a controlled laboratory setting, he will give them a $1 million prize. No one has claimed that prize. He is a good example of the quintessential skeptic. And then the last pillar of the scientific attitude that we'll take a look at is humility, being humble, admitting I don't know everything, and sometimes I'm wrong. I am wrong all the time, and I hope as we go through this course, when you catch me being wrong, that you'll call me on it, and I'll be the first, I hope, to admit with humility that indeed I was wrong, and I'll try to find out what the correct answer was. That's the hallmark of a scientist. So those are the three pillars: curiosity, skepticism, and humility. All right, so a closely connected idea is the idea of being a critical thinker. And in essence, we're going to look at this as someone who demands evidence, not stories, not anecdotal evidence, meaning, oh, this person had this experience and that person had that experience. An example would be, my grandmother smoked cigarettes every day of her life and lived to be 100, so therefore there can be nothing wrong with smoking cigarettes. Well, that's lunacy, and that's not evidence. Anecdotes are not evidence. So a critical thinker demands real evidence to support claims. Replication. What does it mean when we're talking about replication in the realm of the scientific method, this ongoing, never-ending process? If a psychologist publishes the results of a study, let's say it's a study involving motivation in a journal, he or she is going to be very clear in outlining the procedures for that experiment, and hopefully other psychologists, other scientists throughout the nation, throughout the world, following those same procedures will be able to repeat or replicate those same procedures and have the same outcomes. If lots and lots and lots, and I can't give you a specific number, but if many psychologists are coming up with the same results, then we have replication, and it leads to the believability of those results. So let's take a look at the various ways we have available to observe and describe behavior in the world of psychology. And the first one is a case study. A case study involves an in-depth analysis of one individual. So let's say I have a 18-year-old girl who has grown up in a very impoverished home and has become, by her own devices, extremely motivated and successful. And I want to understand how someone can grow up in those kinds of conditions and become very successful. I am conducting a case study. If I want to find out what a large number of people feel about a particular topic, let's say I'm talking about sexual behavior, drug use, cheating on school assignments, etc., I may elect to conduct a survey. And as I pointed out earlier, while you may be able to reach a lot of people, you have to be wary that oftentimes people will lie. Even if it's anonymous, they will lie to themselves and therefore lie to you on these surveys. This is called response bias. Another kind of bias is called non-response bias, and that is people say, oh yeah, you've got a survey, I could care less, and they throw it away. And your only recourse in that case is to go back to those individuals and say, can you please help me out here? So those are surveys. All right, the last one is naturalistic observation, and I'll use the same example I used in our class video. If I send one of you out to the courtyard with a clipboard and a couple of pieces of paper and a pen, and direct you to take close notes on the interactions, the flirting habits between students, and just take copious notes about what you observe. I'm asking you to conduct a naturalistic observation. Why is it called naturalistic? Because you're observing people in their natural setting. It's not contrived. This is not in a laboratory. A laboratory observation, in most cases, is not going to be a naturalistic observation. Okay, let's move on to some more quantitative elements of analyzing data. And the first thing we need to take under consideration is correlation. Is the strength of relationship between two variables. And on this slide you see 6 different possible relationships. And you see again and again the words positive and negative, and that is just referencing the slope of the line. You may be familiar with your studies in Algebra 1 and Algebra 2 of drawing a line of best fit. And if you have a line of best fit through this data, let's start with the top left-hand corner, strong positive. Clearly the line, if you draw through that swarm of dots, would have a positive slope. And notice that those points are closely packed in on that line. So I would describe that as a strong positive correlation. Likewise, on the other side, in purple, we have dots that are clumped around a line with a negative slope, closely clumped, and we would call that a strong negative. And then it goes from strong to moderate to weak to none in both directions. And if I look in the bottom left-hand corner, I see an example of just this random scattering. There's just no way you could place a line either positive or negative on those points. And so that would be an example of a correlation of 0. And by the way, the range of values for correlation, and the variable is lowercase r, is between -1 and 1. So -1 would be a strong negative correlation, and +1 would be a strong positive correlation. 0 would be no correlation. A good multiple choice question on the AP exam might be, what would you conclude if there was a correlation of, let's say, -1.3? Well, your answer would be, that's impossible, because that's outside the range of values of R. Again, the values range from a low of -1 up to a maximum of +1. So -1 to 1. And let's keep in mind that correlation is not causation. It's simply looking at the strength of a relationship between two variables. And when we take the time to do this, we're looking at variables that have the potential of being reasonably related. And here's an example of one that's not, and it's based upon this image that you see of the ice cream cone. And it's taking a look at the correlation between ice cream sales in Miami and crime rate. And what we see is, as ice cream sales go up, the crime rate goes up, which would imply a relationship between the two. Well, if this implied causation, meaning that ice cream sales caused, or an increase in ice cream sales caused an increase in crime rate, a great way to reduce crime would be, hey, let's just stop selling ice cream. Well, that's ludicrous, isn't it? It's ludicrous. And that's an example of an illusory correlation. It's frivolous. It's implying a correlation where none exists. Yes, I can plot these two variables, but there's something called a lurking variable in the background that's explaining why we see this correlation. In this case, that lurking variable that affects both the ice cream sales and the crime rate happens to be temperature. As the temperature goes up, ice cream sales go up, and what tends to happen, especially in areas more prone to crime, when the temperature goes up, is that people's tempers are at a fever pitch. There are more people out and about on the streets because a lot of people in these high crime areas can't afford to turn on the air conditioning. So you've got a lot of people on the streets with their tempers ready to boil over because of the temperature, and they're more likely to become involved in crimes. So that's an illusory correlation. My next point I want to emphasize is that the purpose of correlation, the reason we do correlation studies is to make predictions, and many times these predictions have huge consequences. There have been multiple studies, correlation studies done, and let's say in Africa, taking a look at the relationship between the years of education that a young girl has versus the age at which she first becomes pregnant. Well, that's a pretty big deal, and it turns out there's almost a one-for-one relationship, that for every year that the girl stays in school, she postpones having her first child by another year. So instead of having a child at age 12, if she stays in school 4 or 5, 6 extra years, she may not have a child until she's in her late teens or early 20s. And there we're talking about something that's not an illusory correlation, but it's one of great interest. And again, let me just make the point that we're not saying that correlation is causation, that there's simply an interesting and compelling relationship in that particular case. So correlation is all about making predictions. All right, let's turn our attention to experiments. And what makes experiments unique? Well, experiments are what we have to do if we want to establish cause and effect. The image is of someone smoking, and I chose that deliberately because it's actually an example of where doing experiments was difficult, because scientists were pretty convinced that smoking caused cancer, and to run experiments with human subjects involving one group having a placebo, meaning maybe a cigarette with nothing in it, no nicotine or tar, and another group having the actual cigarettes, knowing ahead of time that you suspected that the cigarettes would cause cancer, is unethical. So they had to use a lot of other techniques to determine cause and effect. But in general, I want you to remember that if you want to establish cause and effect in the world of science, you better start thinking about designing an experiment. So here's an example. Do energy drinks such as Red Bull improve memory? Well, I can conduct an ethical experiment to determine if that is in fact the case. Let's take a look at a way we could design that, and I want you to take a close look at this diagram, because if I was taking an AP exam and they asked me to design an experiment, this is the thing, or the diagram I would first start out with. So let's say I'm starting with 1,200 subjects. What's not shown in this particular diagram, I'll refer to it in the next one, is I have a population of interest. Let's say it's high school students at Palmer Trinity School. There aren't 1,200, but I'm going to first select people from that population, and then I'm going to randomly assign them, as you see in this diagram, into two groups. So let's just say I do have 1,200 people. I'm a big high school. And I'm going to randomly assign them. That's done either with computers, calculators, or random integer tables, which you can find in the back of statistics textbooks. So I'm going to split it up into two groups. 600 people will be in my placebo group, the bottom group, meaning they're going to get something that looks and tastes like Red Bull, but is missing the active ingredients. In the top group, I have the treatment group. They're getting the real deal. They're getting the actual Red Bull with the drug. In this case, the drug is caffeine. And I run the test for, let's say, 2 weeks. And what I would probably do, that's not shown on this diagram, is I would do a pre-test of both groups, checking their memory. Maybe it's their ability to recall strings of numbers. And then I would do a post-test, and at the end of the month or a couple weeks of treatment, and I would then compare the results on both of those. If I chose my sample of 1,200 people randomly from the population, and I found that there was a big difference, I can then say, you know what? Drinking Red Bull will cause an improvement in your memory in this particular population. If I did not use random techniques, I couldn't make that conclusion. And now's a good time for me to emphasize what's going on here with random assignment. Random assignment, when I split those 1,200 people into 600 groups of 600 and 600, the reason I do that randomly is that what it's going to do is cancel out all these different variables that could also affect memory. Things like age and genetics and mental health issues, etc. All those are going to cancel out. There'll be just as many people with memory issues, could be mental health issues in group 1 as there are in group 2, and they'll cancel each other out. Those are called confounding variables, variables that could influence my dependent variable, which in my example here is memory performance. So by splitting them up randomly, all those confounding variables, those other things besides Red Bull that could affect memory, are going to cancel each other out. All right, I told you I would talk about the first step in greater detail, and that is random selection. That's sampling people from your population. You have to have a list of all the available people. This is actually a pretty complex idea. The bigger the population, the more complex it is. But if I was talking about Palmer Trinity, I would have to have a list of all the students in the school or the high school, and then randomly choose my participants from that group. And once I had my participants, I would then split them in this process of random assignment again into a control group or a placebo group that's not receiving the treatment, and then my treatment group that's receiving the treatment or the intervention. Again, notice here's another example of the kind of diagram that I recommend. And here we start with the target population, high school students. I randomly select a group, let's say it's 200, and then I randomly assign them into a treatment group and a control group. I do a pre-test and a post-test, compare the results. All right, let's talk about the two different kinds of variables that we're going to encounter, and then those are the independent and the dependent. The independent variables, what I change in the Red Bull example, what I'm changing is whether or not the person receives a dose of caffeine, and the dependent variable, the thing that I'm observing, is their performance on a memory test of some sort. So the independent variable is something that I change, and the dependent variable is the thing that I observe as a result of making that change. And for what it's worth, the independent variable is almost always plotted on the x-axis, and the dependent variable goes on the y-axis. So make sure you know those terms, independent and dependent, and how they differ. Let's go back and talk about variables. I talked about the Red Bull example with memory. So one of my variables is memory performance. Well, I have to define how I'm going to measure memory. And it could be a string of digits. I'm going to present the participants with a string of, let's say, 20 digits, random digits, and then test them 5 minutes later to determine how many they remember, and that's how I'm going to determine memory. If I was doing a completely different experiment involving, let's say, intelligence, I would have to describe in great detail how I am measuring intelligence. There's all different kinds of ways of measuring intelligence, let alone different kinds of intelligence. So when I ask to operationally define something, I'm asking you to explain how you're measuring that variable. Again, caffeine, I could measure its effect on intelligence or memory, the effect of background noise on memory. So if y'all use the background noise, I'd have to be very explicit as to what I'm doing in this process of testing your memory. What kind of background noise am I using? If I don't specify the kind of background noise I'm using to test someone's memory, then how will another scientist on the other side of the world be able to replicate it? If I run an experiment, and you frequently see this in the world of medicine, that's called double blind, it would mean that neither the participant or the researchers in the field know which is which, meaning whether or not the participant is receiving an actual treatment or a placebo. And a placebo, again, is something that looks like the real deal but isn't. It's a sugar pill, so to speak. A famous example of this involved a large group of middle-aged physicians, male physicians, we're talking between the ages of 40 and 60, who were administered either aspirin or a placebo, and they were measuring, the researchers were measuring the effects on heart attacks, the incidence of heart attacks. So in that case, all the people working for the researchers, the people in the field, the clinicians, as they handed out the medication, either the placebo or the treatment, to the doctors who took part in this, and it was a huge study, they didn't know what the participants were receiving. The only people who knew were the researchers running the show. That's called double blind. The placebo effect is real. Every time you take a dose of Tylenol or Advil, it's at work. A famous example of this that I like to refer to took place during World War II, when horribly injured soldiers, Marines, were on the battlefield. Medics would come up to them, and if they were out of morphine, they would sometimes tell these suffering Marines that I'm giving you morphine. Well, no, they weren't. They were giving them salt water, saline solution. But the Marines didn't know that. And in some instances, when the medics would come back to these wounded soldiers, having replenished their supply of morphine, the Marines would say, I'm okay, doc, give it to somebody who needs it. So powerful is the placebo effect. If you think something's going to work, it likely will. All right, let's turn our attention to more statistical information, and that's measures of center. And the three that you need to be familiar with are mean, median, and mode. Mode you almost never see in scientific literature, but you do see mean and median. I want you to take a look here at these three different graphs. These are three different distributions. A distribution will describe or delineate the range of values that a variable takes on and how frequently it takes them on. So let's look at the one in the upper left-hand corner, and it's a right positive skew. That word skew is important. And you'll notice that you'll see right and left. Well, how do you know which is which? What I tell you is to tell students is to look at the sloping down part. It kind of reminds me of like a horseshoe crab with its tail pointing out to the right. And the direction in which the tail is pointing tells you what kind of skew you have going on. So on the upper left-hand corner, that tail is pointing to the right. We have a right positive skew. On the upper right-hand corner, that tail is pointing to the left. We have a left negative skew. and the one in the center. There's no skew, it's symmetric. All right, let's take a look at the red lines and the green lines in the upper two diagrams. The general idea is as you pull the mean further and further away from the center of the distribution, it is pulled away from the median. Let me explain that a little bit differently. In the upper left-hand corner, as I skew the results out to the right, the mean gets pulled in the direction of the tail. That's what you have to remember. In all cases, the mean, that red line, gets pulled in the direction of the tail. Do you see how in the upper left-hand corner it's pulled to the right, and the upper right-hand corner, that red line is pulled to the left. It's pulled towards the tail. And the median doesn't change that much. So in the upper left-hand corner, that mean is to the right of the median, and on the upper right-hand corner, that mean, that red line, is to the left of the median. Because the mean is really affected by large values, or either very large or very small. And the median is not. The median is simply the middle number. If it's a symmetric distribution like the one in the middle, all three of those lines line up. Okay, we need to have some kind of measure of how spread out our data is, whether or not we're talking about intelligence or scores on a memory test, what have you, and that the accepted measure of variation is called standard deviation. And what you see here is a depiction of something called the normal curve, normal with a capital N. And if you look at the blue section, you'll see, if we consider left and right, that's 68%, roughly. I'm rounding off. 68% of the curve is within one standard deviation of the center or the mean. If I go out to include the yellow sections, I'm going to round off again. So I include the blue and the yellow, and I go out another standard deviation. So now I go out 2 standard deviations to the left and to the right. I now capture 95% of the distribution. This comes in really handy when we look at data that is normally distributed, such as SAT scores, for instance. So standard deviation is a measure of how spread out your data is. If I look at the red curve here, that whatever that number is that describes that standard deviation, I'm guessing that's about 1. And I'll have to draw it on a board to show you how I know it's about 1. But the blue curve is more spread out, is more flattened. It's whatever that number is that describes its standard deviation, that number is larger than the red curve. All right, you saw this graph earlier. I just want to make the point that these kinds of graphs are called scatter plots. And this is how real data looks. It never lines up perfectly in the real world. So this is an example of something called a scatter plot. All right. Let me go back to the earlier example where I talked about Red Bull and the effect on memory. If I determined or I concluded that, gee, the results on these post-tests on memory showed an amazing difference in students' ability to remember numerical information. Well, what do I mean by amazing? Well, in scientific lingo, I would say they are statistically significant, meaning the results were so large they would almost never happen by random chance. Well, what do I mean by almost never? Well, again, using statistics lingo, I would say that the p-value is less than 0.05. Well, what the heck does that mean? It would mean that if I conducted that experiment 100 times, only 5 times out of 100 or less would I see this kind of result happen just by chance. 95 times out of 100, I would expect to see the same thing. So this is the real deal. This is likely, almost certainly, to be true. Statistically significant. Here's some other terms we're going to talk about in order to better describe our results. And the first one is reliability. If my results are going to be legitimate and reliable, the sample that I chose at the very beginning had better be representative or look like the population that I'm studying. So if I'm looking at Palmer Trinity and I'm doing a study on memory, well, I have 6th graders and 7th graders and 8th graders all the way through 12th grade, and I have boys and girls, etc., etc., and I need to have a sample that kind of looks like that population. I wouldn't want to have all 7th grade girls, for instance. That doesn't look like the Palmer Trinity population. So I want it to be a representative sample if I want to be able to reliably conclude that my results can be applied to the population. Another effect on reliability, besides how I chose the sample, is the variability within the sample. And this can be generally controlled by, as you'll see in the next slide, by choosing larger samples. If my sample that I select is really variable, it has a negative effect on my reliability, my ability to make claims about the population, as opposed to minimal variability. Well, that variability is going to even out if I choose a larger sample. Larger sample, less variability, and greater reliability. Does the world and the work that psychologists do in the lab matter? Well, the short answer, as you see here, is yes. This is a picture of an infamous study called the Milgram study, where Stanley Milgram, a Yale researcher, wanted to understand how ordinary people in World War II, he was thinking of the Nazis, were able to commit such horrible atrocities. And so he conducted a laboratory study and uncovered some pretty important principles. And those principles, even though they were uncovered in the world of a laboratory, had immediate connections to what happens in the real world. Does gender matter in our selection of participants in most psychological studies? And it turns out not as much as you might think. It turns out we're a lot more alike than we are different. Now, of course, if I'm talking about a medical study, using the aspirin example, it turns out that gender is pretty important there. Women are not encouraged to take aspirin. But in most psychological studies, it turns out that gender doesn't influence the results as much as you would think. Does culture matter? Same deal. It generally doesn't matter as much as you might think. All right, let's conclude with talking about ethics and how we run experiments on living creatures, be they animals or human beings. The first step, this is really emphasized too much in your book, but I'm going to emphasize it, and that is that before you're allowed to conduct research on human beings, you're required, as I was before I conducted my research on human beings, to appear before an institutional review board, usually comprised of professors at a university who are experts in your field of proposed research. And you have to demonstrate to them convincingly that you have safeguarded the safety, the confidentiality of your participants. It's called the IRB for short. They're going to ask for a copy of your letter of informed consent that you're going to hand out to each of your participants, telling them all the things that might conceivably happen as a result of taking part. In my particular research, I knew that there was the potential for some considerable emotional distress that could come out of it, and indeed many of my participants had an emotional response, but that was in my letter of informed consent, and they all have to sign it before they can take part in the study. When the study is over, when the experiment is over, you're expected to sit down and debrief your participants, and I did that with 11 of my 12 participants. One person was not interested, and this is a good time to point out that I made it clear that all my notes, all my data was completely confidential. That the only person who had access to the names associated with the abbreviations that I used in my write-up was me. Not even my research advisors knew the names of any of my participants. I was the only one who knew, and I made that clear in the debrief. So that's the end of Unit 2, Research Methods in Psychology. Thank you.